Hi everyone, my name is Romário. I'm a PhD student at the Institute of Microbiology here at the Federal University of Rio de Janeiro, and I work with antiviral research. So my thesis project is with new treatment options to treat infection by Zika, dengue, chikungunya, um, and we try to do that by using two approaches. In the first one, we used compounds that are analogs to guanosine, and in the second approach, we use compounds that can inhibit translation initiation. So talking about the first approach first, we use these compounds. They are nucleoside analogs. They are similar to guanosine, but they are different enough to be incorporated into viral RNA and block our viral RNA replication, thus blocking virus infection. And in the second approach, we use these compounds that are similar to our own cap structure there at the messenger RNA. And when you use this compound, it can sequestrate and block the action of EIF4E, which is eukaryotic initiation factor 4E, thus blocking the translation initiation and blocking the viral protein production, thus blocking virus replication. So we have this project together with Professor Wagner Karsten from the University of Minnesota, where I was working with him from 2019 to 2020 because I've got this scholarship from Cap Sprint. So our collaboration started in 2017. We already had this paper published with dengue, where we showed the the antiviral activity of these guanosine analogs compounds against dengue in vitro. And while I was there, I finished the experiments that we started with the Zika project, and now we have this paper published already, where we showed the anti-Zika virus activity of this guanosine-based compound, both in vitro and in vivo. And I also started some experiments using the inhibitors of EIF4E, which is inhibitors of the translation initiation process. But then I could not have enough time to stay there and finish these experiments because of the COVID-19 pandemic. So I stayed there for 10 to 11 months, and I had to come back earlier to Brazil. But our collaboration still is something that's working, and we have a lot of new projects arising, and we actually using these compounds, these inhibitors of translation initiation against the new coronavirus. So we have some preliminary data, and that shows that these compounds can in certain levels inhibit SARS-CoV-2 replication in human lung cells, and we are very excited about that, and we are still working together, as I said before, and we are going to do some new experiments also against chikungunya. We're gonna finish the in vivo experiments for dengue virus, using these two approaches, not only blocking the translation initiation, but also working with these nucleotide analogs compounds.